Intrinsically photosensitive retinal ganglion cells, also called photosensitive retinal ganglion cells, or melanopsin-containing retinal ganglion cells, are a type of neuron in the retina of the mammalian eye. While responses to light in mice lacking rods and cone cells were first noted in 1923, they were forgotten, then rediscovered in the early 1990s. The source of these responses was shown to be a special type of retinal ganglion cell, which, unlike other retinal ganglion cells, is intrinsically photosensitive. They are a class of retinal photoreceptors, excited by light even when all influences from the better-known photoreceptors are blocked. Photosensitive ganglion cells contain the photopigment melanopsin. The giant retinal ganglion cells of the primate retina are examples of photosensitive ganglion cells. Overview. Compared to the rods and cones, the iPRGCs respond more sluggishly and signal the presence of light over the long term. They represent a very small subset of the retinal ganglion cells. The functional roles are non-image forming and fundamentally different from those of pattern vision. They provide a stable representation of ambient light intensity. They have at least three primary functions. They play a major role in synchronizing circadian rhythms to the 24-hour light-dark cycle, providing primarily length of day and length of night information. They send light information via the retino-hypothalamic tract directly to the circadian pacemaker of the brain, the suprachiasmatic nucleus of the hypothalamus. The physiological properties of these ganglion cells match known properties of the daily light entrainment mechanism regulating circadian rhythms. Photosensitive ganglion cells innervate other brain targets, such as the center of pupillary control, the olivary pretectal nucleus of the midbrain. They contribute to the regulation of pupil size and other behavioral responses to ambient lighting conditions. They contribute to photic regulation of and acute photic suppression of release of the hormone melatonin from the pineal gland. Photosensitive ganglion cells are also responsible for the persistence of circadian and pupillary light responses in mammals with degenerated rod and cone photoreceptors, such as humans suffering from retinitis pigmentosa. Recently, photoreceptive ganglion cells have been isolated in humans where, in addition to the above functions shown in other mammals, they have been shown to mediate a degree of light recognition in rodless, coneless subjects suffering with disorders of rod and cone photoreceptors. Work by Farhan H. Zaidi and colleagues showed that photoreceptive ganglion cells may have some visual function in humans. The photopigment of photoreceptive ganglion cells, melanopsin, is excited by light mainly in the blue portion of the visible spectrum. The phototransduction mechanism in these cells is not fully understood, but seems likely to resemble that in invertebrate rhabdomeric photoreceptors. Photosensitive ganglion cells respond to light by depolarizing and increasing the rate at which they fire nerve impulses. In addition to responding directly to light, these cells may receive excitatory and inhibitory influences from rods and cones by way of synaptic connections in the retina. Discovery. In 1923, Clyde Keeler observed that the pupils in the eyes of blind mice he had accidentally bred still responded to light. There was no follow-up at the time. The effect was rediscovered in 1991, when Russell G. Foster and colleagues, including Ignacio Provencio, showed that rods and cones were not necessarily for visual drive of the circadian rhythm, i.e., the body's 24-hour biological clock, implying that a third photoreceptor existed in the eye. Later work by Provencio and colleagues showed that this photoresponse was mediated by the photopigment pigment melanopsin, present in the ganglion cell layer of the retina. The photoreceptors were identified in 2002 by Sama Hattar, David Burson and colleagues, where they were shown to be melanopsin-expressing ganglion cells that possessed an intrinsic light response and projected to a number of brain areas involved in non-image forming vision. In 2005, Panda, Melian, Qiu, and colleagues demonstrated that the melanopsin photopigment was the phototransduction pigment in ganglion cells. Dennis Dacey and colleagues showed in a species of Old World monkey that giant ganglion cells expressing melanopsin projected to the lateral, geniculate nucleus. Previously, only projections to the midbrain and hypothalamus had been shown. However, a visual role for the receptor was still unsuspected and unproven. Research in humans. Attempts were made to hunt down the receptor in humans, but humans posed special challenges and demanded a new model. Unlike in other animals, researchers could not ethically induce rod and cone loss either genetically or with chemicals so as to directly study the ganglion cells. For many years, only inferences could be drawn about the receptor in humans, though these were at times pertinent. In 2007, Zadie and colleagues published the work on rodless, coneless humans, showing that these people retain normal responses to non-visual effects of light. The identity of the non-rod, non-cone photoreceptor in humans was found to be a ganglion cell in the inner retina as shown previously in rodless, coneless models in some other mammals. The work was done using patients with rare diseases that wiped out classic rod and cone photoreceptor function but preserved ganglion cell function. Despite having no rods or cones, the patients continued to exhibit circadian photoentrainment, circadian behavioral patterns, melatonin suppression, and pupil reactions, with peak spectral sensitivities to environmental and experimental light that matched the melanopsin photopigment. The brains could also associate vision with light of this frequency. Clinicians and scientists are now seeking to understand the new receptor's role in human diseases and, as discussed below, blindness, possible role in conscious sight. Experiments with rodless, coneless humans allowed another possible role for the receptor to be studied. In 2007, a new role was found for the photoreceptive ganglion cell. Zadie and colleagues showed that in humans the retinal ganglion cell photoreceptor contributes to conscious sight as well as to non-image forming functions like circadian rhythms, behavior and pupillary reactions. Since these cells respond mostly to blue light, it has been suggested that they have a role in mesopic vision and that the old theory of a purely duplex retina with rod and cone light vision was simplistic. Zadie and colleagues' work with rodless, coneless human subjects hence has also opened the door into image-forming roles for the ganglion cell photoreceptor. The discovery that there are parallel pathways for vision was made. One classic rod and cone based arising from the outer retina, the other a rudimentary visual brightness detector arising from the inner retina. The latter seems to be activated by light before the other. Classic photoreceptors also feed into the novel photoreceptor system, and color constancy may be an important role as suggested by Foster. It has been suggested by the authors of the rodless, coneless human model that the receptor could be instrumental in understanding many diseases including major causes of blindness worldwide such as glaucoma, a disease which affects ganglion cells. Violet to blue light. Most work suggests that the peak spectral sensitivity of the receptor is between 460 and 484 nanometers. Lockley et al. In 2003 showed that 460 nanometers wavelengths of light suppress melatonin and twice as much as 555 nanometers light The peak sensitivity of the photopic visual system, in work by Z.D. Lockley and co-authors using a rodless, conus human, it was found that what consciously led to light perception was a very intense 481 nanometers stimulus. This means that the receptor in visual terms enables some rudimentary vision maximally for blue light.